Hello everyone, I am Shashank Chen. I am studying in first year MSc Chemistry in Jesus College of Arts, Commerce and Science, Ooty Road, Mysore. Today I am going to explain about the specificity and selectivity. Before going to the topic, some introductions of selecting and analytical methods. Selecting and analytical methods involves choosing the most appropriate techniques to accurate and reliably measuring the analyte in a given sample. The selections depends on several factors. The factors are: first one is accuracy, second is precision, third one sensitivity and specificity, selectivity, robustness, ruggedness, scale of operation, analysis of time, availability of equipments and cost. I will explain the specificity and selectivity. First one is specificity. Definition of specificity. Specificity in analytical chemistry is the ability of an analytical methods to measure the analyte and expressively without interference from other substance in the sample. It reflects how uniquely the method can identify and quantify the analyte even in the presence of similar compounds. The next one is methods with high specificity. Mass spectroscopy and nuclear magnetic resonance and immunoassay and chromatography with specific detectors. First one is mass spectroscopy, especially tandem with provide structural informations. Nuclear magnetic resonance NMR uniquely spectra from each components. Immunoassay is antibody antigen reactions highly specific and target molecules, and chromatography with specific detectors, example HPLC and mass spectroscopy, separates and then detects only the analyte of interest. The next one is importance of specificity. Accuracy in diagnosis, example clinical labs, and drugs purifying test, example in pharmaceuticals. Forensics analysis to identify the specific drugs and toxins, and food quality control, example detecting and specific contaminants. Next one is how specificity is evaluated in method validations. First one is analyzing a blank sample and analyzing a spike samples with potential interferencing substance, and demonstrating substance in interference from impurities and degradants, matter components or other similar molecules. Agencies like ICH, International Council of Organizations, and define specific of an validations parameters such that most be demonstrated for identify and purify and assay. And next one is specificity in different type of analysis. First one is qualitative analysis, second one is quantitative analysis. First one, qualitative analysis, specifying the ensure the identify of the analyte is correctly confirmed. And second one is quantitative analysis, specificity ensure only the analyte concentrations is measured accurately, not affected by impurities. The next one is examples of specificity. First one is HPLC, high performance liquid chromatography. It testing a pharmaceuticals and in a tablet formulations using HPLC. Enzymes linked immunosorbents assay. It's detect the HIV antibodies in bloods, and testing must be responsive only for the antibodies produced against HIV, not to other viral and bacterial informations. And next one is gas chromatography and mass spectroscopy. Gas chromatography and mass spectroscopy is detect morphines and urine samples, and gas chromatography separates components, and mass spectroscopy confirm identify the based on unique fragmentations patterns. And UV visible spectra is applicable in food analysis and measuring the nitrate and the concentrations in drinking water using the calorimetry method. And next one is polymerase chain reactions and application in molecular biology and clinical diagnosis. Detect the reacting and PCR primaries and probes and designing to bind only on the viral genetic sequences, and specificity ensure that other coronavirus and human RNA do not giving the false positive. And next one is in IR spectroscopy in identify the aspirin in a pharmaceutical products, and aspirin has specific IR observance patterns such as in a sharp carbonyl peak at 1750 centimeter in and ion selective electrode. This clinically and environmental testing is measuring the potassium ion and blades. And next one is applications of specificity. First one is pharmaceutical industries in the drug purifying testing and stability studies, etc., and clinical and medical diagnosis in and disease diagnosis and etc. Example specific quantities of lithium and blood using ion selective electrodes. And next one is forensic science, drug and toxic identifications and explosive and poison analysis. And next one is food and beverage industries in nutrient qualifications and containment analysis. And next one is environmental monitoring is pollution control for specific detections of toxic substance in air, water and soil, etc. And next one is biotechnology in the protein identification, genetic testings, etc. And chemical manufacturing is chemical for manufacturing is process and monitoring. Next one is selectivity. Definition: selectivity of an analytical chemistry refers to the ability of an analytical methods to distinguish and measure the analyte and in the presence of other components in the sample matrix. It indicates how well the methods can detect the target analyte and without interference from the other substance. Next one is important of selectivity: accurate result and reliable quantifications and quality control. First one is accurate result. Ensure the result are not affected by other components. And reliable quantifications provides truth and worth data even in the complex matrix like biological fluid and environmental samples and or food. Quality control critical in pharmaceuticals and environmental and food industries, ensuring the compliance with standards. Factors affecting selectivity. Spectroscopic method, example UV visible spectroscopy and IR spectroscopy, may have limitedly selective due to overlapping absorption bonds. And next one is chromatographic methods, example HPLC and gas chromatography. IR selectively through separation of components before the detections. And next one is electrochemical methods, can be selective with the proper electrode material and potential control. Next one is instrumentations. Modern instrumentation, example tandem mass spectroscopy and high resolution NMR, and offer high selectively. And sample matrix, the more complex the matrix, example blood, soil, the most challenging it is to maintain selectively. Next one is analytical condition, pH and temperature and solvent and reagent concentration can be optimized and enhanced selectively. How selectivity is evaluated: qualitative evaluations and quantitative evaluations and validation parameters. First one is qualitative evaluation. Check if peak or signals from interference substance overlap with the analyte. And quantitative evaluations spike the sample with known amount of potential interference and assay their effect of the analytical signals. And validation parameters. In the method, validation selectively must be provided according to the regulations, guidelines, example ICH, FDA and ISO. Next one is examples.